Hello, welcome back. All right, our last little mini lesson on the mechanisms of antimicrobial action will be focusing on inhibition of metabolic pathways. So up to this point, we have covered our cell wall synthesis, protein synthesis, plasma membranes, and nucleic acids. And now we're going to take a look at inhibition of general metabolic pathways. I didn't really talk too much. We didn't have a slide because there really isn't too much to talk about on this one, so I'll just mention it here. Any type of drug that's going to block the connection between the pathogen and the host. So it could be like a modified antibody that can bind to the antigen on the surface of the virus or the bacteria, something that would actually bind to the receptor site on the host cell. Anything that's going to affect that recognition or attachment will be able to block the pathogenicity of that particular organism. We just didn't have any pictures or diagrams, just kind of a quick mention of that. So I did. All right, so let's get on to our example of metabolic pathway inhibition. So we're going to focus on this one example, but no, there's many other metabolic pathways out there that can be blocked by these various antimicrobial drugs. So this one is going to focus on the sulfa-based drugs. Remember, those were the first widely used antibiotic was the sulfa-based. And so the three examples that we have here are sulfanilamide, sulfamethoxazole, and sulfazoxazole, all these really hard words to pronounce. And if you take a look, all three of those have that molecular shape in that kind of peachy box. And if you'll notice, it looks like this guy here, which is called PABA, para-aminobenzoic acid, is a precursor for nucleotides, purines and pyrimidines in bacterial metabolic pathways. So in this metabolic pathway here, remember metabolic pathways are these enzymatically catalyzed reactions from a reactant to intermediates to eventually to a final product. So in this case, PABA, affected on by an enzyme, becomes what we call dihydrofolic acid, which is affected by another enzyme which produces tetrahydrofolic acid, which then by another enzyme produces purines and pyrimidines and other nucleotides, which then by other enzymes we know DNA replication and transcription, we get DNA and RNA. So this is the pathway that a bacteria would use to convert PABA into their nucleic acids through this multi-enzyme pathway. Now, in this very first step, this enzyme here is this enzyme here, and it has an active site specific for that ringed structure with the NH2 functional unit on the end, that amine unit, and it fits right nicely here into that active site. Okay, so PABA goes in, the hydrofolic acid comes out. Now, the usefulness of those sulfa drugs doesn't really matter what is out here. So that varies between sulfa drug. What matters is that little ringed group fits into that active site. And so what it does is it competitively inhibits PABA from binding to that very first enzyme in this metabolic pathway. So if we don't get this component of this pathway, we don't get this end product of that pathway. So by putting these sulfa-based drugs in your system, as a cell is actively trying to divide and make DNA and RNA, it's not going to be able to because it's not going to have those precursor molecules that will eventually give it what it needs to do to make more of itself. So this is a great example of metabolic pathway inhibition, but know that there are several others probably doing maybe similar mechanisms, either competitive inhibition or non-competitive inhibition on these enzymes that drive these metabolic pathways. All right, and that's it. So this was a short little video. So we'll finish up taking a look at efficacies. How can we tell how well an antibiotic works? And then we'll wrap up with antibiotic resistance. All right, okay, see you next time.